So yeah, it's a real pleasure to introduce Iain Couzin. He's a director of the Max Planck Institute of Animal Behavior in Constance in Germany. He just told us that he's very close to the Bodensee, so you can make a short walk to that particular beautiful place. He got his PhD in 1999 at the University of Bath, and he's been traveling around. He's had positions in Princeton, University of Oxford, and he also, and that's particularly notable, he won in 2019, got the Lagrange Prize, also together with David Gruber, and he's there in very special company, like Laszlo Barabasi and Mark Newman, and all kind of famous people from complexity science. And so we're really happy that he's here this afternoon, and we're looking forward to your talk. So Iain, go ahead.
Thanks very much. Yeah, it's great to be able to talk to you, albeit not in person. So yeah, today I'm going to tell you about our research on collective animal behavior. So first I want to just give an overview of the department that I run. So it's the Department of Collective Behavior. And what we're really interested in is revealing principles across scales of biological organization. So as you see, we work in a very wide range of systems. And what we try to do is to integrate theory, which can come in different flavors, for example, from simulations where you can look at lots of the complex spatiotemporal dynamics. But also it's very important to employ analytic work to be able to abstract theory and to generalize the results. And then you can see connections across systems that may appear to be disparate, but actually there can be very deep connections. And that's going to be an important part of what I'll talk about today. But theory doesn't make all that much sense in the absence of any data. And so I will also describe how we collect data from complex systems like schooling fish, both in the laboratory and increasingly in the field using deep learning and new technologies. And once one has these types of complex data, then of course we also have to invest in data analysis. How does one analyze these data? How can we make sense of this type of complexity? And so really there's a feedback between all of these different areas, and that's really the ground, the base of what we're trying to do here in Constance. And the theme that connects these apparently disparate systems is information processing. So what separates biological collectives from, say, physical collectives, like materials, is there's an explicit notion of information that individuals have evolved in these scenarios to process information about their environment. So as I mentioned before, because we're interested in general principles, we study a very, very diverse range of organisms, from Placozoa, it's a basal metazoan, literally the simplest animal on the planet, and it's a collection of cells. And here we're interested in how collective behavior has been fundamental in the evolution of multicellularity. And if you track these cells and you look at the density of the nuclei, or you look at the direction here coded by color, you can see these kind of hallmarks of collective behavior that we also saw within the fish schools, these waves of turning within the group. So local interactions scaling to large-scale collective behaviors. Now, when we look at collectives such as these swarm-forming locusts, there's a tendency when you see coordinated behavior to think it's cooperative. But there's a major difference between coordination and cooperation. And in actual fact, these swarm-forming locusts, which some of you will be aware that we had some of the biggest plagues in almost 100 years over the last couple of years in East Africa, exacerbated by COVID, the difficulty to move products such as insecticide and personnel across Africa really made it a terrible situation. And this is going back to my work in Oxford back in sort of mid-2000s. This was the state of the art of the technology. So I developed this software to track the movement of the locusts. And at the time, that was a very important way for us to understand that these swarms are in fact driven by cannibalism. Everyone is trying to eat those ahead and trying to avoid being eaten by those behind. The outcome is collective motion. It's a forced march, effectively, specifically for protein and salt, which is rare in these environments when you've got such large plagues. So that was the state of the art then, this type of tracking technology. But now, using deep learning, with completely markerless. So these locusts actually do have tags on them, so we know who's whom, but now I'll show you shortly, we don't even need those. But the body posture tracking here is completely markerless tracking, and this is focused just on a single individual. So animal behavior is really becoming a very quantitative science. And you can look at these as very rich dynamical systems of the bodies. And I'll come back to that later as well. And so now, instead of just being able to observe and qualitatively describe phenomena from a human's perspective, we can employ a range of methodologies to both track the motion of individuals, but also to use dimensionality reduction techniques from machine learning to understand the principles. And all of the software we develop, such as Trex and Deep PostGIS, is freely available to the community in open source. And so this new software that we've developed is by far the fastest and uses way less memory than the existing state of the art, and allows us to automatically track and identify individuals. So even though the individuals look identical to us humans, the software using neural networks can actually identify them. And this works in pretty much every system we've looked at, from termites to locusts to mammals like mice and rats. And so it's very versatile and it's freely available. And this is important because when you look at collectors, if you're a human observer, you can only really focus on one individual at a time. And really what's happening is a multi-scale processing of information all the time. And so we can, for example, as I showed you a moment ago, we can reconstruct the pathways of light onto the retina using ray casting. So we can actually represent the world from the perspective of the animals. And as you can imagine, if you choose a randomly selected fish near the bottom with the left visual field shown in red and the right visual field shown in blue, this is a complex time-varying scene, both due to the motion of others and to self-generated motion. Yet somehow this little creature has to translate that into adaptive motor control. And what we've been able to do using these technologies is to actually work out which visual features they actually pay attention to in terms of behavior. So a classic hallmark of collective behavior is that as an individual changes behavior, this can be transmitted to others, so-called social contagion. And so what we've been able to do is to use this methodology to work out which visual features are actually causal factors for individuals to change behavioral state. And then we can reveal what we call the hidden networks of interaction within groups, where these edges, the weights represent how strongly individuals influence each other, the colors represent how reciprocal these interactions are, because these are weighted, directed interactions. I might be strongly influenced by you, but you not by me. And in doing so, we were able to show that the network relationships that cause individuals to be most influential and cause the maximum spreading of behavioral change are actually the opposite of those network relationships that cause disease to spread. And so potentially under selection pressure to be able to process information and avoid disease, this is the type of network that you'd want. And it spreads as a complex contagion, which is the same as in human groups. And so, whether you look at humans, birds, or fish, we've all evolved to use complex contagion. That is, you require reinforcement from multiple others before changing your own behavior. Now, I don't want to talk about this study today, so I'm not going to go into further details, but you can read about it in the paper. But this idea of being able to really quantitatively reconstruct the network, so it's not just who's close to whom, which doesn't really tell you anything. It's actually how they're processing the information. So we use the computational visual field reconstruction. We know which visual features to pay attention to, and then it could be highly predictive. We can trigger a behavior in any of these individuals and predict precisely who will be infected by the behavior next and how far it will spread. So this highly complex system has become quite understandable in terms of these behavior spreads. And as I mentioned, these are weighted, directed networks, complex networks. In a more recent study, we were interested in how, when you look at animals in the context of predation, people have looked only at individuals, and individuals will tend to get more sensitive to sensory input when they perceive risk to be available. But in fact, when you start looking at collectives, there's all sorts of other things that can happen. Is it really the individual that's processing information. And we sort of think of the brain as the seat of knowledge, of information acquisition and computation. But these brains are embedded within this network. And so now it's not clear where the computation arises. Does it arise within the individual brains, or does it actually arise within the topology and the structure of the network of communication? And again, just to summarize work over the years, because I want to move on to some new stuff today. And here you can see this beautiful visualization. This is a real fish school with these networks, and it's, you know, they're not scale-free networks. They're not small world networks. They're in their own unique network class that's been under very strong selection. So lots of very interesting things about the network topology. And we found that computation is an emergent property of the structural properties of the network in a wide range of contexts, such as sensing long-range environmental gradients. The individuals cannot sense the gradients, but the group can sense the gradients via the network of interactions, via regulating social contagion, as I mentioned a moment ago, as well as modulating behavior in response to increased risk. The individuals do not change their sensitivity to sensory inputs. What they do in the presence of risk is they change the topology of the network, and it's the network that then changes the signal to noise ratio and processes information about the changing environment. And, you know, it might sound quite group selection-y, you know, but, you know, I specifically almost always work on individuals that are unrelated to each other. So we can really ask questions about how do unrelated individuals evolve these types of emergent properties. And, you know, we've also done modeling and experiments to really show how easy it is for selfish individuals to evolve these types of collective computation capabilities. And it really does draw a parallel, a direct parallel, to how processing occurs in other types of collective systems like the brain, where each neuron is, you know, relatively sophisticated, but certainly the networks of communication give rise to higher order computations. Okay, so I'll talk about some well, oldish work, or pretty old work first, because it really sets the scene for some of the most recent unpublished work I would like to discuss today. So I'm also showing you pictures of fish because I happen to have these nice pictures, but you can think very generally of, you know, birds, fish. We've even done studies with humans. And later on, I will show how this generalizes to primate groups and bird flocks. So you can think very generally about this. The idea here is that, you know, how does leadership and collective decision-making occur within animal groups? How do they make informed, unanimous decisions? You know, does this individual that has information, for example, it knows where a food source is, or it knows parts of our migration route, does it need to signal to others? Oh, sorry, that's my dog is just trying to get in the room. Let me just let. Come on. You know, does it need to signal to others that it has information? Do others need to recognize, oh, this individual is informed, this individual is smart, and we follow it? Well, there's no evidence of that in most of the systems we study. We also do study signaling, by the way, but I'm not going to talk about that today. Do others need to recognize each other to know who has information and who does not? Again, we have no evidence in these systems that that's common. And so the individual that has information has a goal-oriented vector, a desire to move in a given direction. But in order to maintain the benefits of group living, it also needs to reconcile that with its social tendencies. And so in this model, we assume that individuals are not aware whether others are informed or uninformed. They're not explicitly aware. They can't count. They start at random positions and random orientations, and they just have simple local rules of attraction to other individuals, or avoidance if they come too close. Now here, I'm going to show you individuals with a preferred direction of travel in white, but that's only so we can see it. In the model, the colors are just for us humans. In the model, there's no way of inferring whether others have information. And so these individuals have a desired direction of travel, this goal-directed travel. And what we find is that without signaling and without individual recognition, this information can be conveyed very accurately within the group. So we don't need complexity of individual at the individual level for this to occur. And so if we look at this preferred direction of travel, and we look at, say, 1 or 5 or 10 informed individuals and a group of 100, and the green cross just shows you the centroid of the group, One individual is not able to convey information to others. 5 informed individuals, after an initial period of reorganization where they occupy the frontal edge, they kind of convey the right information. The group sort of moves towards the right, but not very accurately. But with 10 informed individuals, as I showed you a moment ago, it's near spontaneously and very accurate. And so if we normalize, or if we normalize this, so we have 0 is no information transfer, 1 is perfect information transfer. And I should note that these are all stochastic models. The individuals have got sensory and motor noise. You can see for all group sizes, the accuracy increases very steeply as we increase the proportion of informed individuals before plateauing. And if we look at, say, 85% accuracy, so arbitrarily, for a small group like a group of 10, we need around half of the individuals to have that information. But if we look at a large group, like a group of 200, then it's less than 5% of the individuals for that same level of accuracy. If we simulate migrating groups of millions or cells, for example, where you can have millions or tens of millions of individuals migrating, you can actually have an infinitesimal proportion of individuals that actually know where to go, yet it looks to the observer as if they all know where to go. Oh, now you want out. Sorry, everybody.
Quite unpredictable animal behavior.
Exactly. I'll come to I'll come to how predictable animals are in a moment. And so, not just happy with just modeling, we also have done a range of experiments where we've tested these ideas, including with humans. But here's an experiment we did with fish, where the red individuals were informed and the blue were uninformed. Fish have amazing memories, by the way. This whole 3-second or 10-second memory span that you can read about in the popular press is complete nonsense. Within 7 trials, they remember extremely well and they'll maintain that memory for months or years. And we could show that you have the steep increasing accuracy as a function of the number of informed individuals within the group. Now, so far, my informed individuals have all been in agreement with each other about where to go. But just as in human society, these animals live in fission-fusion groups. Then they're not always with the same individuals. They will mix and merge with other individuals. They have no knowledge of the information content that others have. So in this case, they don't know if anyone else agrees with them or disagrees with them. So again, simple local rules of interaction sort of melded with this goal-oriented behavior. So without the ability to count, starting at random positions, random orientations, with these very simple interactions, can a group actually come to a collective consensus decision? And so now what's going to turn out to be important is not just the number of individuals with different preferences, or the strength of those preferences, those are both important, but also explicit the geometry of the problem. These individuals are moving through real space. And the angle at which you want to move through space is going to turn out to be critically important to explain animal behavior. And so I'm going to keep one informed subset always to want to go along 0 degrees, along the x-axis. And then I'm going to model a group that disagrees with them. It might disagree by a very small amount, and that's this top dotted line. Towards the left there could be 10 or 20 degrees, all the way up to 180 degrees, where they actually want to go in exactly opposite directions. And I'll show that these exact numbers don't matter in a moment, but here I'm going to have a group of 100 where 5 prefer to go one way and 5 want to go in a different direction. And I'll run thousands of simulations for each angular difference, and we'll look at the probability distribution of where the group goes. So the group is not splitting, the group always chooses the same direction. And so what we find is what's called a bifurcation, whereby below a critical difference of opinion, the group will spontaneously adopt the average opinion, even though no individual knows whether anyone agrees with them or not. And even though we have 90 individuals in this case, 90% of the group doesn't know anything. But as the group, imagine the group's going towards a red target and a green target, red to the left, green to the right. As the group approaches that angle will inevitably increase, and it will reach this pitchfork bifurcation, where now the group has a choice and has to decide to go one way or the other. And we find that they spontaneously, 50% of the time, the whole group will choose the green direction, and 50% of the time, the whole group will choose the red direction. Now, what if we have a difference of opinion? So I'll just show you a model here. So here there's equal number that wants to get to the white and the red targets, and the green are the uninformed. and it's going to be random which one they select. But what if I add just one extra individual to the groups? Again, starting at random positions, random orientations, you've got no way of knowing if anyone agrees with you or disagrees with you. So we have 6 versus 5 now, and these are noisy local interactions, as you can see from the animations. And what we now find is that, as before, below that critical difference, they will spontaneously move in the average direction until they get close enough to the targets and you reach the bifurcation. But now, instead of it being 50-50, that tiny, tiny little difference in this noisy system with no ability to count breaks the symmetry, and the system will move almost always in the majority preferred direction. It's extreme sensitivity at that point, which I'll come back to the reason for that later on. So they can vote on the move. So when you look at fish schools and bird flocks, really they're mobile computational entities. And when I did this work, back in 2005, we sort of wrote in the paper, isn't it amazing they can do this despite all of these naive individuals, these uninformed individuals? And it was only in 2011 that I realized that it's actually because of the uninformed individuals that they're so capable of this. But just to show you some animations again. So here we have, again, an equal number that wants to get to the white and the red target. This is the video I showed you a moment ago. But I've now added one extra white individual. So just one extra individual. And the colors, again, for our benefit, will almost always reach the majority target with no ability to count or anything like that. And so what I want to now go on to, I've always been fascinated by this idea of collective minds. To what degree, when we see collective computation in the natural world, can we think of the sort of computation that we think about in humans? Is this just an analogy? And what I now believe, it really goes beyond the metaphor. I think there are deep fundamental mathematical principles at work in the brain that really reflect the processes that I just showed you. And that's what I want to talk about today. So now I'm going to move from the group as being the collective, and I'll come back to that again. Don't worry, I'm going to come back to collective behavior. But I'm now not thinking about the group as being my collective. Now I'm thinking about the individual brain, the neural groups as being the collective. So how the neural interactions scale to how an individual behaves. So it's still collective decision making, but it's within the individual organism, and it's now with neural groups. And so to study this, it's incredibly difficult. So we had to invent a new technology, which we call holographic virtual reality for freely moving animals. So I always, I've tried to develop robots to work in, in animal groups, and the animals are always too smart, or the robots produce vibrations and so on. Or it's really hard to have 100 robots that you're controlling with real animals. But using virtual reality, we can really do some pretty amazing things. And so it's based on something called the anamorphic illusion. Such a powerful illusion that I can tell you right now that this is not a three-dimensional object. It has no volume above the table. Yet, even if I tell you, your brain believes it to have a volume. It's such a powerful illusion, and you can see it broken now. And even if it goes back into roughly the same position, your brain will spontaneously pop it back into having a depth, having a volume above the table. Similarly, this training shoe does not actually exist. It's not actually in 3 dimensions. And so if one actually renders a computer scene from the perspective of, in this case, the camera, you can actually move behind and interact fully with rendered objects using a conventional screen. So the objects can be any distance to the animal, even though the screen, of course, is a fixed distance. And so we've developed this technique for many animals, as I'll show you, but this is just showing in fish with a virtual fish below and a real fish above. We project onto a bull, correcting for distortion. We track the animal in 3D with very high precision and high frame rate, very low latency. And this world looks a bit weird. This is sped up. This world looks a bit weird to us, but from the zebrafish's perspective, that pillar it's there in the tank with it, and it will avoid it as if it's there, even though, of course, this is just an illusion. There's nothing really there, it's just light. We can also create virtual fish that look exactly and move exactly like real fish. And then we can have virtual fish populating the same water volume, the same tank as a real individual. And so here you can see the virtual fish is just moving in a simple circle, but the real fish believes it to be there in the tank with it, even though the projection surface is only the edge of the ball. So it's a really powerful illusion. Now, there's one problem here, which is that these are different virtual reality tanks for the fish. But if I put more than one individual in the tank, the world will look distorted to the second. You have to do it exactly perspective corrected from the eyes of one individual. So I can't put multiple individuals in these tanks because the world will look distorted to the second one. But what I can do, and what we do, is we can network these systems together such that the individual in the closest tank is interacting live in real time with a hologram, a holographic representation of the individual in the second tank, the third tank, and so on. And so now we call this the matrix for obvious reasons. So now you can see two juvenile zebrafish interacting not in the same physical world, but in the same holographic world. And analyzing their behavior in great detail, looking at their turning speeds and their forward speeds, there is no difference between their behavior in the real world on the left and the virtual holographic world, or the matrix, on the right. So we have a really powerful approach whereby we can, and we have 6 of these systems now. Here, just 4 of them networked together, so we can have individuals interacting. Of course, we can have arbitrarily complex environments. It's a video game engine. We can have virtual predators. It's really up to our imagination. And we can also play around with things like geometry. Geometry need not be Euclidean in a virtual world. We can put in time delays to understand how animals deal with space and time when they make decisions. And as I mentioned a short time ago, we also do this with other animals, such as Drosophila, which is a model organism in biology, and also with swarm-forming locusts that I showed you at the beginning, moving on this motion-compensating sphere. So as the animal moves, the sphere moves such the animal's always on the top, so it can move as far as it likes. So lots of advantages, like extreme control. I mean, we can give every individual the same information, the same part of the retina. We can quantify into individual differences. We can scan parameter space and really connect our theory with our experiments in a way that's really impossible doing conventional experiments. So using this technology, I want to come back to this question of how do neurons within the brain act as a collective, and how do they make decisions, and what does that tell us? What can I bring from a study of collective animal behavior to the study of neural interactions? And so this paper actually, as of today, is now in revision at PNAS, but it's already available on the bioarchive. So if anyone's interested, you can find that paper and you can go into more details. So now the individuals are not organisms with different preferences, but they're neural groups that are competing with each other to decide for the brain where to go. But like I showed you before when I looked at animal groups, the angle, as the animal moves through space, the angle will change, the representation will change. And so I had this instinct that, all of this work I've been doing on animal collective decision-making could actually help us explain how the individual brain makes decisions. And even though, I mean, there's tens of thousands of studies of decision-making by individual animals, I couldn't find a single paper where I could get the trajectories of the animals as they made decisions. So to look at this feedback between the geometric representation and the movement decision, which then impacts the geometric representation, which impacts the movement decision, and so on. And so we developed a very simple model, which sort of gathers the basic information about how the invertebrate and the vertebrate brain behave, whereby we have reinforcement or excitation of positive feedback among neural ensembles that have a similar directional representation, and then either global inhibition or negative feedback, it doesn't really matter, that dampens a sort of negative feedback in the system. So very simple, again, interactions inspired by our studies of collective dynamics. And what the model predicts is, for a wide range of neural noises, that if the animal is far away from the opposite, just as we saw with the animal group a moment ago, then the brain should spontaneously average the vectorial inputs and choose an averaged direction. However, this is only up to a critical point at which the symmetry should be broken, we predict, and that the brain then chooses one versus other option. Okay, and if the options are identical to each other, it's going to be 50-50 which one the animal chooses. And so if we think about this as the animal moves through space, the prediction is that the animal will move through space and the brain will average the inputs until a critical point, at which point the animal will choose one or other option. And if the options are equal, it will be 50-50. Now, this relates to the study of complex systems or phase transitions generally, because one universal feature of a phase transition in any system is that at the critical point, the property called the susceptibility will increase dramatically. And the susceptibility, if you look at, say, a magnetic system, a ferrous magnetic system, you know, if the system is completely disordered and you apply a magnetic field, it's hard to get those particles to align. On the other hand, if the system is already extremely ordered and magnetized, and you then apply a field to try to change the direction of those particles, you're going to have to apply a strong field to change them. However, at that tipping point between disorder and order, the system becomes maximally sensitive to external inputs. Now, of course, this is not a magnetic system, but the concept is exactly the same. So a system at the critical point here will be susceptible to any differences between the remaining options. So any differences between spatial targets, for example, their contrast or something like that. And you don't have to put that into the sensory system. It's an emergent property of any complex system undergoing a phase transition. And so modeling this, you know, if the animal is able to go through this critical point, we start the experiment such that the animal moves and hits the critical point, it can exploit that susceptibility to make very accurate distinctions between options where only one or two neurons difference between the preferences. It's very much like one extra individual wanting to go to one target versus the other. Here, the brain spontaneously becomes very sensitive to any differences in preference between the neurons. So, of course, you know, theory is great, but we also want to test this idea. And as I mentioned, you know, we couldn't find any data at all where people had actually asked, how do animals move through space when they make decisions? And they often start the animals directly in between the two options, which is beyond the bifurcation point. So the theory, as I showed you before, predicts this bifurcation as animals move through physical space. So using virtual reality, we can test these ideas. We've done so in the classic model organism, the fruit fly. And what I'm showing you here is the probability distribution of many trajectories of the fly moving towards, not the fly, hundreds of flies moving towards the target. And because it's hard to really see the patterns in the trajectories because they overlap, I'm going to show you the density plots. But these are based directly on the raw data. And again, all of the data is available, and you can see in the supplement of our paper all of the raw information. And you know, if this is a general feature, this should occur in other brains. So the locust is a widely diverged insect. They're not really related to each other. And again, the locust also shows this bifurcation. So the brain appears to go through this critical state when going from averaging to decision-making, which could be hugely advantageous. But what about three options? If you look at neurobiology and psychology, almost all experiments are done with two options, because computationally it's difficult. But believe it or not, you know, animals live in worlds with three or many options, and these options are distributed in physical space. Again, we couldn't find any data from people exploring this basic idea of how do animals move and make decisions. And what's kind of cool about the theory, again, coming for free as an emergent property of this very simple model, is that we predict that the brain spontaneously breaks the world down into a series of binary decisions. And this really surprised me. I would have thought that the neurons sort of pointing to the rightmost option would cancel the ones to the leftmost option, and the system would just go to the center and be quite boring. But in actual fact, you know, if you actually model this, you find that the system spontaneously creates a bifurcation where it excludes one of the two extrema, and then you enter, you then hit a second bifurcation point where the system will decide between the remaining options. And close to each critical bifurcation, the system becomes extremely sensitive to differences among the remaining options. So what a wonderful emergent property for a collective decision-making system to have, because that sensitivity, that ability to discriminate between options comes for free via what we know about any system that undergoes a phase transition or phase-like transition. And so to experimentally test this, it becomes very hard, but again, VR allows us to do so. And again, we find evidence in the fruit fly and in the locust that the brain indeed exhibits this double bifurcation as they move through space, making their decisions. And to my knowledge, this is a behavior that's not been seen before. And the cool thing about the model as well is that although it's experimentally very difficult to test more than 3 options, these experiments are very, very hard to conduct. The model works seamlessly for any number of options in the environment. And so these properties are very generic. So I'm trying to sort of get at this point that, perhaps this is a principle of decision-making across scales of organization, exploiting the bifurcation, exploiting the susceptibility. But I've looked at 2 insects, and even though they're divergent from each other, it's still the insect brain. What if we go back in evolutionary history and we then think of a completely different brain, the vertebrate? And so choosing zebrafish, again, a model biological system, we can study their dynamics. And again, we utilize our virtual reality. So here we don't have static options, like discrete options in space, like targets that the locusts and the flies wanted to move to. Here we change the ecological context to have the virtual fish here shown in red, just so we can see, are actually moving through space. And we ask, where does the conspecific, a real fish, follow? And we predict, again, as we change the distance between these virtual fish, we will reach, they'll average, and adopt a central position until a bifurcation, a very sharp bifurcation, where they'll choose one option or the other. And again, our virtual reality is the only way that you can really test this type of idea. And indeed, we find very strong evidence that in the brain of the animal, there is indeed a bifurcation as predicted by our theory. And we can again look at 3 options where the double bifurcation looks a bit different now because of the moving frame of reference. But if you look at the data, you can again see evidence for this double bifurcation, again, as we predicted. So this property seems to occur across scales of organization, and really is the same computational type of dynamic that we saw within our moving animal groups. And so now working with Armin Ball, who's just moved from Harvard to Constance to set up his own group, he specializes in whole brain imaging of zebrafish. We're now actually studying the networks within the brain to find how this is actually implemented and to test our ideas of neural integration. And we've also been able to go back to our studies of animal groups and to a completely wildly different model, the model of our swarms and animals moving, and yet that model makes exactly the same predictions for exactly the same reasons. So even though these models differ vastly in the microscopic details of how they're implemented, the fundamental principles occur because of this basic excitation or positive feedback combined with inhibition or negative feedback. So can this actually also help us understand decision-making in the wild? So working with Ari Strandberg-Peshkin, who's my PhD student at Princeton and is now a group leader here at the Max Planck Institute, and Damien and Meg Crowfoot, who's one of the other directors at our institute, we asked this of wild baboons. So now we put GPS collars on almost all of the individuals within the group. And for obvious ethical reasons, we did not put collars on juvenile individuals. But now every second, we get the location of these individuals as they move through space. And when I looked at these data, the colors here represent just males, females, and subadults. Turns out that none of that status doesn't matter, the sex of the individual doesn't matter to the story I'm telling today. But I found tens of thousands of examples where some individuals would go one way and others would go in a different direction. And yet the group, again, to maintain the benefits of group living, would come to consensus. So we can look at these examples, as I said, tens of thousands of them, and actually test this bifurcation theory in the wild with the baboons. And if there are equal number wanting to go each way, or if there's a majority wanting to go one way, you can see even a small majority, one extra individual, breaks the bifurcation, and the individual will choose the majority preferred direction. So exactly as we found within our virtual reality experiments with these simpler organisms. And this is a very complex animal in a very complex landscape. But we found that the dominance status of the individuals didn't matter, their experience didn't matter, their sex didn't matter. It really all comes down to this, this collective dynamic. We've also taken these ideas and tested them, or these types of ideas looking at leadership and so on, and tested them within free-flying storks. These are real data again, these are GPS and accelerometry data. And you may wonder, you can see some of them flapping in this thermal. You may wonder how we know if they're flapping. Well, we have the accelerometer as well as GPS. And the accelerometer gives us the wing beats of the animal. And then when the animal is gliding, we can tell that it's gliding. So we can get lots of information. And we use solar powered GPS, so we can actually track these animals over their entire lifetime. And so we catch them near constants, and then we can actually track them as they move across sub-Saharan Africa during their annual migrations. And so it's just a wonderful time to be doing this type of science, because we can get this sub-meter resolution of every wing beat. We can then see how they're moving in the thermals and be able to even reconstruct how the thermals are buffeting them. We can ask, how do they make decisions to move between thermals? Because you can't see where they are necessarily, but you can exploit the information provided by others already thermally. And again, we found that there's this small number of informed individuals that guide the group through this process. And so we can look at these different strategies in the natural world. But it's pretty expensive and difficult to do these studies in the wild. So just before COVID, I had the great fortune to go to South Africa with Bill Hansen, who was the vice president of the Max Planck and the president, and we were doing a study on buffalo. And as you can see, this involved us having a helicopter, having a whole team. We had to have vets, we had to have Paulie with his gun there to help protect us from interested wildlife. So it's very, very difficult and time consuming to do this type of study. And we also wanted to sort of democratize this and to go beyond GPS and to do massively multi-scale imaging of ecological systems with simple off-the-shelf consumer technology. And we actually started this with our studies of baboons using a relatively expensive drone to actually image the three-dimensional structure of the landscape. But now it's possible to do this just with off-the-shelf drones. And so we can reconstruct to about 2 to 3 centimeters spatial resolution the three-dimensional structure of the landscape through which the animals move. And then working with Blair, Ben, and Jake, who are 2 postdocs and a PhD student, this landscape looks two-dimensional to you, but this is actually a three-dimensional landscape. And these individuals moving through it are actually an endangered species of zebra called the Grevy's zebra. There's only around 2,000 of them left in the world. So the animals that are most critically important for us to save are the ones that we're least likely to get permission to dart and to put collars on, because of course that could impact their behavior. So here using multiple drones, and it's sped up, by the way, you can see the little tails wagging, plus our deep learning body posture technology, we can now get very detailed information about how the animals behave and move through space. And so we can, I just want to sort of end my talk today to just to tell you a little bit about what's happening in Constance, in the hope that now COVID is hopefully receding and we hopefully won't get too many dangerous variants, that it might be possible for some of you to come to visit us. And so when I came here 6 years ago, there wasn't even an undergraduate course in behavior at the university. So I've been doing a lot of building. And so one of the things we did was there's this excellence strategy in Germany. And this provides large sums of money for up to 14 years to really focus on specific research topics. And so this comes in 2 periods of 7 years, and we have the first 7-year period at the moment. And this is great for a small university like ours because it really brings researchers from across the campus to work together on collective behavior. And so we have biology, computer science, economics, psychology, physics, social science, and mathematics. So a very diverse community, predominantly of young scientists. And we also got this, happens to be the same amount of money, but a different funding scheme. Again, a competitive funding scheme from the German government for a new science building, because if we really believe in complexity science and interdisciplinary science, and yet you go to universities and you still find the biologists are in the biology department, the chemists are in the chemistry department, physicists are in the physics department. Well, we need a building where we can actually put all of the people together to work together. And that building, actually, I should have a new picture because it's actually opened today. And I should have had a new picture of this. And underneath the building, in the ground, we have a huge animal facility, including what we call the imaging hangar, which is a 15 by 15 by 8 meter space where we can do sub-millimeter 300 hertz tracking, even of insects. Now, it's a really unique, globally unique space, and it's open for anyone to use at no cost. So it's really for the global community, not just for us. Another reason to come and visit us is this is the view from our new building. It's absolutely stunning across Lake Constance towards the Alps. So I really hope that if you're interested in this, you'll come to visit us. But because we didn't know how to track animals at this type of scale, when I first moved here, we renovated an old barn. This is what the barn looked like when I started, and this is what it looks like now, where we have this sophisticated imaging technology. So we've developed a whole range, whole suite of different tech, as I said, that allows sub-millimeter precision, 300 Hertz tracking, even now of bees or of locusts in these types of environments. So we can look at how animals are seeing the world. And so we're getting lots of cool scientists coming from all around the world to come and use this facility. And in the new building, this is a photo of the new facility we have, the imaging hangar, which scales us up. It's 4 times the volume of the existing space, but we use similar technologies. And not everyone can afford such amazing facilities. And so we also want to democratize these technologies by using that fancy tracking technology to then train neural networks to do the 2D to 3D transformation. So you can just use webcams to approximate the same data. And we also collaborate with psychologists, for example, at the University of Constance, who are interested in collective eating behavior. So the system can really work from insects all the way up to humans. And we also have a new Max Planck Institute of Animal Behavior. The building is just, we've got the funding for this. And these are the 3 directors. There's Meg Crowfoot, Martin Mikulski, and myself. And we've got 60 million euros. And that building will hopefully open in around 2026, 2027. And so that will provide extra capacity for this type of research. So Constance is a really fun, exciting place to be. So please do come to visit. And thanks a lot for listening.
Yeah, thank you very much. Really nice. It's great to see all this fantastic development, and fantastic to see all the animations and all the that there's no animal safe anymore to get tracked in the world. Fantastic. Yeah. Okay. I guess there will be a lot of questions, and you can post them in the chat. I don't see anything.
I will quit my talk because I can't actually see the.
Yeah. Okay.
I can't see the chat while sharing.
Yeah. Well, I can start. I was, of course, intrigued by that what appears to be a pitchfork bifurcation, which you called, right, in this sort of choosing different directions, and also you mentioned later in the brain. That suggests that, I mean, there's an underlying continuum, right? Maybe where you can describe this kind of interactions. I know from people who've done, partial differential equation formulation of flocking, for instance, and you can come up with a PDE theory, right, of flocking.
Yeah.
It's sort of integral differential PDEs.
Yeah.
I mean, in the theory you use, is that individual based, or is it also more continuum based? And do you see those kind of pitchforks also in that continuum description?
So that's a really good point. And so I simplified it. Again, I said the paper, as you know, is on the bioarchive. And so you can see that again, in that paper, we used a model of spin systems.
Yeah.
And we also developed an analytically retractable version of that model. So we can really analyze the nature of these bifurcations.
Yeah.
And in previous work, we've also employed things like coupled oscillator models,
okay.
Where the direction of travel is really a phase in an oscillator. And there are differences actually in the nature of the bifurcation depending on the formulation that one uses.
Yeah.
In the coupled oscillator formulation, we found it was a monkey saddle bifurcation. In the spin system model, in actual fact, it's a Hopf bifurcation.
Okay.
The Hopf bifurcation.
Yeah.
And so the question then becomes, well, that's very, very interesting to sort of make a prediction regarding those bifurcations. But what can that tell us experimentally? And one of the cool things about the Hopf bifurcation is that it has a sort of oscillatory capability. And I thought that's a bit bonkers. I was working with Nir Gov, who's at the Weizmann Institute, is a chemical physicist working on this. And he then predicted that the animals will oscillate between the two options. Now, of course, animals can have a stopping rule. When you reach somewhere, you can stop to feed. But what if the animal can't reach anywhere? It has two options, but can't quite reach. Well, it turns out people have done this experiment with flies. And what the fly will do is just perpetually go back and forth all day. And again, if you look at our fish, and we look at how when we have that bifurcation, it's not like they just sit in one branch and stay there. They actually regularly oscillate between the two options. On average, they're in those branches.
Okay.
So that type of modeling suggests to us that they're exhibiting a Hopf bifurcation, and that could be important. And this is still speculative. It could be important because it's a very nice way in its optodynamical system sense. Once you get somewhere, and so, I've always been interested in decision making in terms of, once you make a decision, but what if it turns out not to be a good one? We always think about just making a decision, then it's over. But in reality, you make a decision and you want to change your decision potentially to go to the next best option. And this type of bifurcation allows you to spontaneously do that. The animals will spontaneously then move to the next best option, just again based on the dynamical principles of the nature of the bifurcation. So by natural selection, by tuning the way that these local interactions scale to the collective properties, in principle, could tune the type of bifurcation that's exhibited in order to allow it to have these adaptive features. Now that is speculation for now, but at least the data are consistent with that hypothesis. And so this is one of the reasons we want to go into the brain to actually look at the neural dynamics directly, because in the virtual reality, because it's a closed loop system, once the animal is at the critical point, if it's really moving through the critical point very quickly, then we can't really study the critical point. You can maybe see the brain dynamics exhibit, say, scale-free correlations, and then they go through. But we can also just have the feedback loop causing the animal to remain in the critical point. So it can't get out of the critical point. And then it should be very sensitive to perturbations. And we should also be able to at least allow the collective dynamics of the brain to be in that critical point for much, much longer than in the real physical world via this trick of closed loop control. And there we hope to be able to really understand whether our predictions about criticality are indeed playing out in the neural dynamics.
That also was my second point, because for a pitchfork is a very special bifurcation, right? You need symmetry. And for Hopf, that's sort of that's a generic bifurcation, even in case without symmetry. So that's more likely to occur in that sense.
I mean, this is exactly what we think is occurring now. I mean, we used to think it was pitchfork, then we thought it was monkey saddle. Now we tend to think it's probably Hopf bifurcation. So we're doing a bunch of studies now to look at these types of, because so far I've just showed you the sort of the average outcome. But of course we also have the dynamics, we have the motility, the motion of the individuals through the whole process. And so we're now, again, working with Neogov and others, we're really looking at those dynamics to see if they can inform us and allow us to distinguish between different hypotheses. Also, we can break the symmetry in different ways, experimentally using the VR. And so that's kind of currently what we're working on.
Yeah, very, very ingenious. Yeah, really nice. Any other questions? I don't see anything. Yeah, I see Kate. Go ahead. Kate, can you unmute?
I could also read, if she prefers, I'm very happy to read her question. Just put a question.
Yeah, you can do that. Yeah, maybe you do that. Yeah.
Oh, her
microphone's not working. Okay. Shall I do it?
By all means.
Yeah. So she says, very interesting talk, thanks. I was interested in what you mentioned about the baboons. If I heard correctly, was that only collective dynamics are important in determining direction of travel, rather than social dynamics such as rank. Is it possible that different informed individuals have different amounts of influence over the collective decision making? If not, how and why do you think that the animals are able to overcome the influence of rank in this decision making? Yeah, interesting.
Yeah, it's a very interesting point. And so, I was, one of the reasons I wanted to work on the baboons is because they are a classic organism for hierarchical structure. And it's a linear dominance hierarchy. All males are dominant to all females, and it is a linear structure. And so, but I get frustrated when I watch, television shows and they say that the dominant male is dictating everything that they do. Because those dominance ranks come from aggression among individuals, like who wins in the aggressive encounters. And that's all about mating. That's all about access to females and mating. Now, when you're looking at animals moving through space, it's not clear to me that that should actually directly relate to who's influential in terms of making movement decisions. And so what I hoped to happen was that we would discover a hidden hierarchy that's unrelated to, or partly related to the dominance hierarchy, but is separable from it. What we actually discovered was that there is no hierarchy in terms of the movement decisions. And the dominance rank could explain, I don't know, by 4 or 5% of the variance. It was very, very low. And this kind of surprised us at first, but it opens up two potential hypotheses for why this might be the case. One is that when the group actually does make its decision and gets to the food source, the dominant male in particular, but the dominant individuals in general, can monopolize the food source, definitely. And so potentially the dominant male, has evolved to allow the system to have this democratic consensus decision-making because it facilitates collective intelligence, and he can then exploit that because when they actually make the choice, if it's better than his own choice would have been, he gets the benefit of a great choice, plus he gets the food that's associated with that great choice, or preferentially gets that food. That's one hypothesis, and a non-mutually exclusive hypothesis that I didn't have time to talk about much today, but we've explored extensively in a paper in Science in 2011, is that when we have the strong difference in influence between individuals in terms of their leadership capabilities, that exerts itself only when you do not have uninformed individuals. Putting uninformed individuals that don't care what's going on, spontaneously make the group democratic, i.e. they remove the strength of preference. Even though individuals still have a strong preference or weak preference, dynamically it can no longer play out. And so I would, so one possibility is that the dynamics of these systems, just by having individuals who are uninformed or have low preference strength, spontaneously democratizes the decision. So that paper is called Uninformed Individuals, I think like Democratize Consensus Decision Making or something like that. It was published in 2011. And so I don't know which of those two hypotheses, if either, really explains what we see, but they're both consistent with what we see in the baboons.
Oh, sorry, any response, Kate? Okay.
She says thanks.
I also thought that the dominant male individual was dominant over every aspect of life, but interesting to know that it's not necessarily the case. Okay, good. Yeah, good to hear. Okay, then there's a question by Jork. Can you unmute and ask it?
Yes, hi. Thanks, Iain. A really interesting talk. So if these decisions on group movement in the baboons, so say, for example, with regard to foraging, so if they can rely so much on group knowledge, what would these kind of data bring forward within the discussion about social intelligence versus ecological intelligence in primates?
So I think it's a really interesting question, because if you look at the literature from, say, insects, social insects, as group size increases, the insects get dumber and dumber, i.e. like army ants, which is the largest groups, they're literally blind, the African army ants, the biggest groups on the planet. And they really use collective intelligence to solve all the problems, and it means the individuals can't even feed themselves. They're so reliant on others. If you look at in primates, as group size increases, the brain volume, certainly of the regions of the brain that deal with social cognition, increase in size. And it's always been thought that this relates to the social complexity, the complexity of life, maintaining third-party interactions and third-party relationships and so on. We do have also examples in fish where fish are capable of understanding third-party relationships and so on, and those fish also have relatively large regions. And so I think, really what's playing out here is, these collective dynamics allow the system to make highly effective decisions. We've even been able to show that they're very close to provably optimal decisions that would be very hard for any individual in the group to even do. But it also frees you up. I mean, anything that frees you up, because the brain is such an expensive organ. Each cell is 10 times more even than a muscle cell to run. Anything that frees up cognition to do other things could be useful. And while they're on the move, they're doing all sorts of other stuff. That potentially does involve social cognition. They're checking out each other. They're having little fights. They're having scraps and so on. And so it's possible. Again, we don't know by any means, but it's possible that you've got this really multi-layered cognition. That the individual cognition is very, very important for certain aspects, like really understanding the depth of social relationships. But then you can kind of outsource the spatial cognition of the group to a large degree, not completely, but to a large degree, by using these types of collective processes. And I think that's kind of really one of the most exciting things that I would like to study is, putting the cognition into collective cognition. It kind of surprised me. I wanted to start studying more complex animals because I was kind of bored of these sort of self-organizing models. I wanted to see what extra do you get when you have a cognitive entity. And at least in terms of these movement decisions, it's not that important, but there's tons more that these animals are doing. And so it's a very rich social dynamic. And I think potentially it frees them up to focus their cognitive attention. And also, as I mentioned before, anything that you can allow the collective to sort of solve for you reduces your need to run your brain, which is going to be selected for as well.
All right, thanks.
Thank you.
Okay, any other question in the chat? I don't see anything. Okay.
I have a question, Hank.
Oh, yeah, Robert, yeah, go ahead.
Yeah, sorry. At the beginning, you spoke about coordinating and cooperating behavior, and a lot of the stuff you were talking about was seemed to be goal orientated. But what happens when there's a predator and the goal is it's very self-orientated goals? So it's, maybe the individual is trying to survive. Do you see any coordinating to try isolate specific individuals in the groups that they're targeted, or is there any difference between animal groups cooperating and coordinating behavior?
Yeah, I think that's, again, really good points. And predation has been one of the strongest selection pressures on the types of groups that we study, especially these small fish. It's estimated around 92% mortality through predation in the first few years of their life. So extremely strong selection pressure. And we've studied that in a variety of ways. We studied that in the lab, where we've very recently, we've got a paper in review right now, where we've got real predators, hunting individuals in groups. We've done studies in the field using new types of radar, imaging, where we can actually track predators hunting within groups and look at the collective properties. And we've also done experiments where we've had virtual prey being hunted and selected on by real predators. So real predators hunting virtual prey, and the virtual prey all looking identical, but evolving their behaviors. And so there's a variety of things we find. One important thing is that the individual fitness is so highly integrated with the fitness of others due to these social relationships that and that the system properties are so important that the collective effects are really critical. So, for example, in the wild, the predators will even hunt in groups. They'll form lines to slice through the fish schools, because it's only if they can get the fish schools small enough, these prey schools small enough, can they break down the ability for these prey schools to transmit. Those beautiful videos I showed at the beginning where the waves just almost instantaneously propagate across the group. Well, that really allows individuals to sense and detect and respond to predators that they can't see. And we looked in very turbid environments where the interactions were really local, and yet these prey groups were just dancing around the predators unless the predators managed to slice them up. And if they manage to slice them up, then the information transfer is still as fast, but it's over a shorter range. And then if you look at the relationship between per capita risk and group size, it's incredibly steep. Once the group gets big enough, the predators just can't catch anyone. So these collective effects are really critically important for these organisms. So even though the animals are unrelated to each other, there's a mutualism here. They're mutually benefiting from these the sort of collective intelligence that exists at the level of the group. And when we've done virtual reality experiments, we find that it doesn't just pay to evolve to be close together. You have to evolve to be also exhibiting aligned movement. So even in the absence of detecting predators and responding to predators, even if you remove that from the equation, they will still evolve collective motion, and that's associated with the predator finding it much harder to target individuals. So there's multiple factors at play that sort of have selected for highly coordinated movement and dynamics in the face of predation. And as I mentioned also briefly, the way they even change their sensitivity to risk. The groups get more twitchy and sensitive when predators are around. And that's actually not mediated by individuals changing their sensory processing. They just change their group structure. and that spontaneously, that then emerges. And so the individuals are really doing a heck of a lot of their processing regarding risk and predators using the sort of network of interactions because of the capacity for them to effectively feel a predator over very long distances and respond to predators over very long distances, which would be impossible just using their own cognition.
Yeah. Okay, thank you very much. Anyone for a last question? I don't see anyone. So then let me close by thanking you again, Iain, for a fantastic talk. It was really fantastic to see what's going on in this field. It's also fascinating, also the technology you're using. It's incredibly impressive. So,
well, thanks a lot. It's been a lot of fun.
Yeah,
thanks very much for the invitation.
Yeah, you're very welcome. So also to anybody who's still present, thanks a lot for attending. And we have a few more activities in June, right? There's a COVID symposium, and then there's also Andrea Rinaldo is also talking in June 17. And so we'll hope to see all of you again, and have a nice rest of the day. Thank you, Iain, again.
Cheerio, everyone. Bye.
Yeah, bye.
Take care.